Hello and welcome everyone to Red Planet Live. I am your host, Ashton Zeth. I'm elated to be hosting the Mars Society's podcast and leading the conversation about human exploration of the universe and the future settlement of Mars. As a longtime space enthusiast, I am passionate about STEM education and making humanity an interplanetary species. Thank you everybody for joining today and supporting Red Planet Live. So as usual, before we kick off the conversation, just a couple Mars Society announcements. Want to remind everybody that today is Giving Tuesday. So this Tuesday, we invite you to support the Mars Society in advancing the exploration and settlement of Mars. Your donation directly fuels vital research, educational programs, and initiatives like the Mars Desert Research Station, helping to prepare for humanity's future beyond Earth. Every contribution, big and small, brings us closer to making humanity multi-planetary. So let's make this Giving Tuesday truly out of this world. Visit marssociety.org/donate to contribute today. Secondly, are you looking for the perfect gift for the space enthusiasts in your life? This holiday season, explore the Mars Society's online shop. From apparel, accessories, to collectibles, there's something for every space lover out there. Not only will you find some unique gifts, but every purchase also supports the Mars Society's mission to advance the exploration and future settlement of Mars. So visit themarsstore.com today and give a gift that's truly out of this world. All right, last announcement. Do you have someone in mind that would make an amazing guest for the Red Planet Live podcast? We're always looking for passionate experts, innovators, storytellers who are shaping the future of space exploration. So drop us your suggestions and let us know who you'd love to hear from. We are typically booking several months out, so share your suggestions now and help us bring the best voices in space to the conversation. All right, now for the good stuff. Today, we have a special guest who is Dr. Stephen Benner, a distinguished biochemist at the Foundation for Applied Molecular Evolution, who is renowned for his work in synthetic biology, dynamic combinatorial chemistry, and paleomolecular biology. He was the first to synthesize a gene for an enzyme and developed artificial genetic systems that have enhanced clinical testing for HIV and hepatitis. His paleomolecular biology work has allowed the study of ancestral proteins in extinct species. Benner earned his bachelor's and master's in molecular biophysics and biochemistry from Yale University and his PhD in chemistry from Harvard University. He has also received prestigious awards, including the Dreyfus Award and Sloan Foundation Fellowship. Everyone, welcome Dr. Stephen Benner to the podcast. Excited to see you.
Good evening.
Looking forward to chatting with you. I mean, that intro, so many just amazing experiences there. And I got to say, I've never said molecular biology so many times in a single sentence. So you win that award too.
Well, remember, I'm getting old, so we have a lot of years.
Hey, we've still got time. We might add a few more to that list. But for today on the podcast, I do a segment called Question of the Day. It's meant to just be an icebreaker. I promise no right or wrong answers. We won't judge you. It's just your opinion. So for today, the question is, if you could name a new city on Mars, what would it be?
Oh my goodness. Yes.
I pulled that out.
Yeah, I mean, obviously, one tries to make these cities reflect somebody who was excited about Mars. You know, you can't fault someone like Percival Lowell. Okay, so he is a man who probably was wrong about most of what he said about Mars, but he did make a lot of charitable contributions to the activity of Mars, and for the longest time, right, he was driving the view in culture that Mars could possibly hold an intelligent biosphere. So let's make it Lowell City.
I love it. That's a good answer. That's a really good one. For being put on the spot,
wrong answers. So
yeah, exactly. No wrong answers. But for being put on the spot, that's a pretty good one. I like it. Well, I want to remind everybody, if you have any questions for Dr. Benner, please put those in the chat. I will try to sprinkle those out throughout the conversation. But want to utilize our time. I know we've got a lot of those people that are listening, so let's dive right in. But typically, we like to start the conversation at the beginning. So, Dr. Stephen Benner, if you could tell me a little bit about like your background. Obviously, we heard about your educational background and your bachelor's and your master's from Yale and your PhD from Harvard. But take us farther back. how did your interest in science start? Did you always want to be a chemist when you were a kid? How did you get on this path?
Well, it's hard to say. I mean, there's lots of influences. I mean, as a small child, I was involved in rock collecting. My older sister collected rocks. And of course, it's great fun if you're out there camping like my dad used to like to do. And we would go around to various places and pick up rocks. A lot of good places, in the United States and Canada for amateur rock hounding, as it's called. So
Mhm.
you can go to Bancroft, Ontario, for example, and you can find sapphires, black sapphires, and a rock outcropping in the middle of the woods. If you go to North Carolina down the Skyline Drive, there are places where you can find emeralds. And if you go into the Cowie Valley around a place called Franklin, which is just in the western part of
Mhm.
the state, you can find rubies and garnets and various other things. And then, at that point, you get to be very much familiar with the chemical elements, right, which go into the rocks that you're finding, and some of them are quite pretty, right? So they're very attractive, and you can still go to. I'm outside my office right now. We have a small museum which contains a lot of the rock specimens, but also elements, samples of pure elements like hafnium and beryllium and ruthenium and tungsten and in small cubes. So that was, of course, very much a big part of it. But then you got to keep in mind, as a chemist, almost all chemists at some point in their youth are enchanted by fires and explosions. So a lot of chemists end up where they are basically pyromaniacs whose activity has been diverted towards socially productive things. So it's a combination of enjoying things explode, but then trying to understand why it explodes. And so yeah, at that point it was really pretty much set. I had a chemistry set, and then I was doing magic shows as a teenager to fifth grade birthday parties where you would bring science into some of the illusions that you were creating, and it was downhill from there.
I'm gonna have to add magician to your introduction next
Yes,
time. We're gonna add that.
yes, I
Do you
still do.
still collect rocks? Are you still into pyrotechnics? You like fireworks still as an adult?
Yeah, well, of course, most of the fireworks I see are staged by other people on Fourth of July, but because what a professional fireworks guy can do is a lot better than what you can do privately. So, yes, but sure. I mean, I still have a weakness for fossils and rocks, and so when my kid is growing up, we would go on fossil hunting digs and rock finding places. I don't know that he took anywhere near as much of an interest as I did, but yes, the answer is we're still enchanted by this. And of course, Mars, a lot of rocks there, and a lot of what goes on as we try to evaluate Mars for its habitability, how life would have originated on Mars, how it would have survived the cooling and drying of the planet. A lot of that ties back to geology. So, as
Mhm.
a chemist, I'm constantly using geology background to talk about what goes on Mars and how life would have emerged there. Because remember, the emergence of life is very much a discussion of how chemistry, organic molecules in particular, interact with the rocks that are in the environment and produce things that are useful, maybe even things that will support primitive genetics, and from there, of course, primitive Darwinian evolution and primitive life. So yeah, it's all one big happy thing, and there is no biology, and certainly no alien biology, separate entirely from the geological environment of the planet where you're hoping to find life.
Mhm. I'm by no means a scientist or an engineer. That is not my background. But I did enjoy geology in college. That was one of my favorite classes. And every so often, I get to pull out a party trick, which is like, which kind of rock is this? Igneous, metaphor, for the everyday average person that is not into science quite like we are. It's
So you
it's a
don't
fun.
like watching things burn?
I mean, who doesn't? Who
Okay, all
doesn't?
right.
I mean,
Just asking. As long as
there's a
it's not
few
in the kitchen.
candles back there.
I do.
Um,
Yes. Okay. Yeah.
yeah, there's a couple back there. Okay, yeah. So we talked about, you hinted at Mars, and of course we're gonna get into that. But I would say, like, just to start, for those that are a little bit unfamiliar, maybe we could talk about your work in pioneering the creation of artificial DNA structures. Like, for those that don't know, can you clarify, what that means? How do you create synthetic DNA, and what does that mean
Sure.
for our understanding of life.
Well, yeah, I mean, so obviously there is DNA as we teach it to 10th graders in high school, which is a four-letter alphabet that forms a double helix
Mm-hmm.
held together by base pairs, which are A pairing with T, adenine pairing with thymine, G pairing with C. This is something that Watson Crick came up with in the 1952, 53 frame. It's older than I am, but we're taught in school that this is an elegant, sometimes beautiful, perhaps unique way to support the informational needs of life, right? So, what is the core of molecular evolution is to be able to make copies of something where the copies have imperfections, mutations, and then be able to copy the daughter molecules so that the mistakes are also then propagated. So if you have replication with error, where the errors are also replicable, that
Mm-hmm.
is the essence of the molecular process that we call evolution. And you are, of course, then going to superimpose on top of that natural selection so that the mutations that help you acquire more resources from your environment, help you select a mate better, help you reproduce, all of which are part of this concept called fitness, right? Natural selection has to then select from the random variation that is part of the process of replication, those variants or those mistakes that give you more fitness.
Yes.
So that's a molecular basis for DNA and RNA that has the molecule we teach in 10th grade, has these bases that are forming the pairs that are holding the information by their sequence or their order in the DNA molecule, being held together on a backbone, which has a repeating negative charge. Phosphoric acid is with the phosphoric acid linkage, hence the DNA. The A is acid. Deoxyribose is the sugars that are holding the phosphates together, and the bases are all put together. The N is the nucleic acid nucleobases. So, yeah, so the question that arises is that, in fact, the only way you can do it. So, if you go to Kronos and meet a Klingon, or go to Vulcan and meet Spock, will they also have exactly the same structure? Is that the only way you can actually store information in a replicable form where the replication with errors can be then followed by further replication of those errors? Now, the answer to that is, if all you know is what you do in 10th grade biology class, hey, what else would you know? But once you get a little bit more advanced, you can do synthetic organic chemistry, and that involves making new molecules. So you can start to ask, what if and why not questions, where you say, well, why are the backbones phosphate? Why not something else? And why are the sugars ribose? And why not something else? Or why are the bases A, T, G, and C, adenine, thymine, guanine, cytidine? Why not something else? And so the synthetic chemist can go back and say, hey, if I go to Vulcan or Kronos or Mars, and if the life there has a natural history that is independent of the natural history that we have had on Earth, would you still have the same DNA? Now, it turns out that on Star Trek and the voyage through the galaxy, most aliens sort of look like Hollywood actors with prostheses, maybe. And you can have Mr. Spock marry Mrs. Spock and have a half human, half Vulcan offspring. Of course, what part of the word fiction don't you understand? But that's part of the issue. But yeah, so it turns out that the Star Trek imagination is not terribly broad in terms of producing alien life forms, but the chemist can be. And so in 1986 or so, obviously, the most important thing in science is, Ashton, do you know?
Oh, I
Money.
would say Mars always,
No,
but
no,
you
it's money. It's money. It's money to do no bucks, no Buck Rogers, right? You have to be able to do chemistry. So I moved to Switzerland because, of course, it was absolutely crazy in the United States to try to raise funds to
Mhm.
ask what alternative DNA molecules might Martian life have. But in Switzerland, they were very much forward looking. They actually understood the potential of these crazy questions and leading to stuff that's good. So we moved
Mhm.
to Switzerland and spent 11 years there and built the first examples of a DNA molecule with, instead of 4 letters, we've got 6. and then we have 8, and then 10, and then 12, and then we had to worry about how to get those molecules to be copied, and developed a whole bunch of chemistry to support that process, and then copied with errors. And then the question is whether they would copy with errors, whether you could get them to evolve to do useful things. So what happens when you try to do that is you have a lot of failures. A lot of the molecules that you designed to be replacements of DNA actually don't work very well or don't work at all. After a while from doing that, you have some successes as well, and you then start to, as a chemist, understand what kinds of molecular structures are able to support Darwinian evolution and therefore life, and what kinds of structures are not able to support Darwinian evolution and therefore are not able to support life. And this is all laboratory experiments. We don't leave Earth, but now because of this 30 or 40 years of experience developing these alternative genetic systems, we sort of know what is possible out there. So right now we're supported by NASA as part of one of its interdisciplinary centers for astrobiological research, which is looking at where you might look for life on the planet Europa, or the moon Europa, which is one of the moons of Jupiter, or whether you could perhaps find life in Titan, which has got these oceans of natural gas, frozen methane, and whether they would be able to support Darwinian evolution, and what kinds of molecules might support replication with errors, where the errors are themselves replicable in a methane ocean. And of course, Mars right now is, and from a point of view of cost effectiveness, it's the easiest place to go. It's the most like Earth. Whatever life is there is more likely to be recognized as life than if we go to some place that is truly inhospitable from our perspective, like Venus, but which could still support biological molecules. But Mars is a place to go right now to actually seek extant life. And there's a lot of that reason to believe that there is life living today in the near surface on Mars. So, but because we have done all the explorations in the laboratory about how life might work chemically, even if it does not share a natural history with Earth and does not actually have Earth-like DNA, we know what kinds of molecules might work as alternatives, and we know how to look for them.
So speaking of Mars, given your research that's related to Mars, how do you see synthetic biology contributing to the search for life, or the terraforming of the Martian environment?
Okay, well, those are two very different things. So the searching for Mars, what synthetic biology does is let you ask what if and why not questions. So what if it were silicon instead of carbon? Well, they're good answers to that. So what if there were 6-letter DNA as opposed to 4-letter answers? And of course, synthetic biology has provided you insight to possible answers for that question. Or what if the DNA molecules on Mars are not joined by phosphates by something else, or have a different kind of sugar? And then you also do research in parallel into the origins of life. So you say, what kind of chemistry would have occurred on Mars? It's privileged. That is chemistry that could not have happened on Mars, given the geology and the solar radiation and the atmosphere and the amount of water, this type of thing. And so from that, you're coming together with a model of the universe, and that's a technical term. We're not talking about the universe, but a universe of chemical possibilities that might be encountered for life on Mars. And that's now, then you go to Viking 1976, you land in duplicate life detection missions on Mars. They produced results. Then you have to ask yourself the question, now that you understand sort of the universe of possible biological systems that might exist in that environment, might have originated in that environment, how would you interpret the Viking results to be evidence for life or evidence against life? And that synthetic biology has informed that discussion, so that in 1976, we think they got the answer largely wrong. But, in 2026, 50 years later, you are able to say that synthetic biology, as well as a lot of studies of, natural biology here on Earth and extreme environments in particular, are allowed, lets you say much more intelligently how you would interpret the Viking 1976 experiments, what life there might look like, how it might earn a living, what kinds of metabolism it might have. And of course, that's informing you on how to look for life. Now, so that's the exploration of Mars. It also is telling you, if you send Elon Musk or the Chinese National Space Agency or NASA puts humans on Mars, we have to worry about how they would interact with a potential biosphere there. Part
Mhm.
of that is to worry about how we would do what Matt Damon did, is basically put his poop on the surface of Mars and obscure our ability to find indigenous life. Of
Right.
course, and
Contamination.
so that's now that doesn't address your terraforming question. I have on the wall behind me a Mars flag, right, which is red, green, and blue. Where the blue we put the oceans back on the surface of Mars, and so yeah, it's going to be that's an engineering problem. That's
Like,
really not a chemistry problem.
Dr. Benner, can you tell us or share some reasons why we may think that Mars has a near surface microbial biosphere?
Yeah, I mean, so yeah, so I'm not sure how. I mean, I'm sure your listeners are familiar with the Viking missions.
Hmm.
The Viking missions did 3 experiments that were all intended to find life on Mars. The one that's most easy to understand is from Gil Levin and Patricia Stratt, and they put radioactive food under the surface of Mars and looked for the exhalation of radioactive carbon dioxide. So if I were to feed you radioactive food and you swallowed it and you inhale a little oxygen, you would burn the food, get energy out of it, run across the marathon, and exhale radioactive carbon dioxide. So that was a label release experiment. It's a label because the carbon 14 is a radioactive isotope of carbon that labels the carbon in the food instead of carbon 12, which is not radioactive. So you have a hard time detecting not radioactive carbon dioxide in a Viking experiment manifold, but it's very easy to do so if that radioactive carbon dioxide has labeled carbon that is giving off high energy electrons that you can detect once it's exhaled from the Martian surface. Now, the result known to almost everybody in the listenership is that Gil Levin and Patricia Stratt saw the evolution of radioactive carbon dioxide. They saw the metabolism of this food. And that was interpreted at the time as a life positive sign. There was a second experiment where they're looking for photosynthesis, basically. They're putting radioactive carbon dioxide. They also put a little bit of radioactively labeled carbon monoxide
Hmm.
because the Martian atmosphere has a both. And they put them above the Martian soil in the ratio found in the Martian atmosphere and looked for what we call technically the fixation of carbon. What that means is the same thing that plants do. The plants are out there in the sunlight. They get some energy from the sun. They get a
Hmm.
little carbon dioxide from the atmosphere, and they reduce the carbon dioxide. That is, they put hydrogens onto the CO2 and make organic materials, and that's plant stuff.
Hmm.
And that's, of course, the food that you eat as a as a plant eater. And of course, you're a secondary and tertiary plant eater if you eat um, uh, animals. But the point about the experiment was to go to the bottom of the Martian food chain on the surface of Mars and see if they could observe the fixation of carbon dioxide into organic molecules in the Martian surface. This is an experiment designed by Norm Horowitz, who was a professor at the time at Caltech, and they saw fixation of carbon dioxide. Okay, so that they saw organic molecules in the surface. So the first thing that they saw was label release. That's biologically called respiration. You inhale and exhale
Hmm.
the carbon dioxide when you eat radioactive food. And then they saw carbon fixation, which is the bottom of the food chain that is based on higher organisms able to do respiration. So the third thing they did was gave them their first unexpected result. They expected to see the surface exchanging gases. So they put a bunch of gases above the surface and they put a little moisture, and they are assuming if there is some life in that soil, they would see the release of gases or the fixation of gases. And what they did was they moistened the soil and they immediately saw the release of oxygen, O2, like you and I breathe.
Hmm.
Now, they did not know quite what to make of it. You could view this as the other half of photosynthesis, because when plants fix carbon dioxide, they also release oxygen to the atmosphere. That's where we get the oxygen, the 21% oxygen that we have in the atmosphere of Earth today, what we breathe. Um, and so they were a bit puzzled by getting that oxygen out just by manhandling, moistening, treating abusively from the Martian perspective of the soil. They were surprised to see that.
Mhm.
So you can say we have three life detection experiments. Two of them are clearly positive. One of them may be positive. Why is it that you think that they didn't find life on Mars? And I mean, actually, do you think I can ask you that question. When you were
Is that a
getting taught,
personal opinion, or not
well, I mean,
officially?
you were taught something, right? And if you took a standard course or read a standard textbook on Mars, they will say that the life detection experiments failed to find life on Mars. And I've just told you that two and a half of the experiments succeeded in finding life. And so the question then becomes, why is it that the textbooks say differently? Now, there's a history here. And the history is that after they had done those three experiments, they did a fourth. They had an experiment. They put down a machine called a gas chromatography mass spec instrument. So they scooped up some soil and they were looking for organic molecules in the soil.
Mhm.
They put it into a little cup. They heated the cup to 500 degrees centigrade. It was at stages. They hoped, therefore, to volatilize all of the organics that were in that soil. They blew it into a gas chromatography column, which your reader need not understand. Your listeners need not understand it, but basically it separates various molecules, gaseous molecules that might be emerging from organics. And if I heat you up to 500 degrees centigrade, 1,000 degrees Fahrenheit, you emit gases also. But then they then look for the masses of these, and so the report is that what they detected was only methyl chloride, a little bit of methylene dichloride, and some freon. And so they said, wait a minute. Well, they had run the machine flying out to Mars. So they had detected freon in the machine. Freon is a fluorinated, it has fluorine and carbon in it. It's used as a
Mhm.
refrigerant. And it
Yep.
was clearly a contaminant brought with them from Earth. But they had not seen the methyl chloride in the flight out to Mars. The methyl chloride was new on the Martian surface. That puzzled them. They did not know where they would get methyl chloride. And so they assumed that the methyl chloride was a contaminant from Earth, even though they had not seen methyl chloride in the flight from Earth to Mars.
Mhm.
So already you're in a little bit of a logical problem here, but what's very amusing if you actually read the papers is that was what was reported, but then you have a report in Science magazine. Now you got to keep in mind Science magazine is the creme de la creme. It is the magazine where you are saying your research results not for somebody in your field, but for the entire community. And so
Mhm.
in Science magazine, methyl chloride ends up appearing in the table not as something new on Mars, but it's in the table as something that is a contaminant from Earth. There's no
Mhm.
explanation for it having been moved there. But then when Norm Horowitz wrote his review in now Scientific American,
Mhm.
so, right, Journal of Geophysical Research was the first journal, which is for the specialists. Science is for the science community, but Scientific American is for people like you, right, and my mother, right? So, what they say, what Norm Horowitz says in the Scientific American article is that they found nothing on Mars but cleaning solvents brought from Earth. Well, Norm has confused chloromethane with dichloromethane
Mhm.
because he's a biologist, doesn't know the difference. Methyl chloride's a gas. It's not a cleaning. It's not a solvent at all. It's not a liquid. And so all of a sudden, you see now in the literature, you see a mutation of the reports of scientific results. This is not genetic mutation, but this is information mutation, whereby you go from not having three biological experiments, which at least two and a half of them are in favor of life, to all of a sudden losing the results because the GC mass spec was interpreted as showing nothing in the Martian surface by way of organics, except as it came to be reported, organics that were brought as contaminants from Earth. So now you have Jerry Soffin, who is running the whole mission, and he says, well, that's the ball game. No organics, no life. Why? Well, because the only way we think that we can implement Darwinian evolution, including you and me, is if we have organic molecules, right? And so you can't find any organic molecules in the Martian soil, there can't be any life. And it's
Mhm.
worse than that. Organic molecules fall onto Mars all the time. There are things called carbonaceous chondrites, carbonaceous, meaning they contain carbon, they contain organics, they are meteorites. They fall to Earth all the time. I have some of them out here in my little museum, but they fall on Mars all the time. So one certainly expects to see organics on Mars from meteoric infall. In fact, Norm Horowitz had criticized the gas chromatography mass spectrometry experiment by saying all you're going to detect is the organics that are coming in from meteorites. They're not interesting. They will obscure the biological organics from the life living on Mars. But they didn't see those either. Now, my only contribution to this whole thing was a paper we published in 1999 saying that even if the Mars Viking experiments had been sitting on a pile of meteoritic organics, they would not have seen them. And of course, Gil Levin and Patricia Strott pointed out that the GC mass spec was not sensitive enough to detect the life in the Atacama Desert or in Antarctica, which would have produced a positive result in their label release experiments. But never mind, because they found no organics, nobody at the time discussed the sensitivity problem, and no one was aware of the chemistry that we knew. But by the time we got around to publishing in 1999, that's a quarter century after Viking, and so by then the culture has come to the conclusion there are no organics in the Martian soil, therefore there can be no biology in the Martian soil. It got worse that the oxygen that was released from the Martian soil had to be coming from, in this view, a strong oxygen in the surface that was burning the organics. Any organics were there. And that, in fact, that strong oxygen was what was oxidizing the food that Gil Levin, the labeled food, that Gil Levin and Patricia Strott. So the whole story became Martian surface is strongly oxidizing. There are no organics in the soil. And by the way, that persisted until a couple years ago. I mean, Martian rovers have gone to Mars in 2002, 2003, 2004, 2010, and so on. There are a lot of them, Curiosity, Opportunity, Spirit, they're wandering around the surface, and they're finding organics all over the place.
Mm-hmm.
But the culture is so strong that when Vicki Meadows and Heather Graham are putting together a meeting, a workshop, as they did 3 years ago, to try to come up with what they call community standards, to decide when a life detection experiment at a certain standard and therefore should be published, when someone should be allowed to claim that they have identified a biosignature. They got a whole committee together. Vicki and Heather ran it, and so they generated a report saying you shouldn't publish or shouldn't allow certain things to be published based on they're not meeting these standards. But they justified this whole set of standards by pointing to the Viking results and how they were misinterpreted as being possible signs of life. And that was a bad thing. Well, of course, they had not read any of the literature, or at least were not cognizant of it, as far as we can tell in the report. At least it did not make it into the report. There are organics all over the place on Mars. There's the potential for life everywhere. And in fact, the problem with the Viking results was not that they were too eager to assume that the biosignatures were signs of life. The problem with the Viking story is that they were too eager to interpret signs of life as not being life. And so we're right now back to the question, well, how would you interpret these experiments? Now, of the experimental results from Viking? Well, the answer is you would first try to put this into a geological context, and you would ask yourself, if I were a microbe, how would I handle my environment? Well, the answer is I'm scarce in water. By the way, I'm scarce in oxygen. The atmosphere of Mars has a little oxygen, but not much. So if I'm a photosynthetic organism fixing carbon during the day, that's Norm Horowitz's results. The question that we ask, if you're a sixth grader is, well, what do the Martian organisms that are doing photosynthesis do at night? Well, what do the plants outside my office do at night? Well, the answer is they respire. They take the food that they make during the day and they make CO2 and release it to the atmosphere. But of course, the Martian life has a disadvantage compared to the Earth life. On Earth, if I'm a plant and want to respire at night, I can always find the oxygen to pull from the air to burn the organics that I made during the day. But on Mars, there's hardly any oxygen in the atmosphere. So the Martian organism, if he's going to make organics during the day, they would like to store the oxygen for overnight metabolism. And so this is bacterial autotrophs, as we like to say, respiring with stored oxygen for overnight metabolism. So when they put the water on the soil in the third experiment and saw saw oxygen release, what they're seeing, according to this particular model, is the release of the oxygen that the Martian biosphere is storing during the day, so that it has something to respire with at night. So you're seeing Gil Levin's experiment, the label release experiment. The third experiment, you're seeing the respiration. So you've seen the fixation of organic molecules. That's Horowitz's experiment. You expect that a Martian microbe in that environment would store the oxygen for overnight metabolism. So that's you would be releasing when you dump water on it, like, you know, you're basically upsetting the organism. It's releasing the oxygen. And of course, the respiration is Gil Levin's experiment. So in fact, all of the Viking experiments are consistent with bacterial autotrophs respiring with stored oxygen for overnight metabolism. And it turns out that bacterial autotrophs respiring with stored oxygen for overnight metabolism has the initials BARSOOM, Barsoom, which, if you're a fan of Edgar Rice Burroughs and John Carter and the Princess of Mars, you know that the Martian, in his fictional story, call their planet Barsoom. You probably didn't know that. You're too young. Right?
I don't think.
Do you even know who Edgar Rice Burroughs is? He's the guy who invented King Kong. So that work is much more better known than John Carter, Princess of Mars. But yeah, so the point is that the Viking experiments are all consistent with a microbial life form that would be earning a living doing what you and I would do if we were going to design a microorganism to live in that ecosystem.
Well, you hit on a question that we had. An audience question was about the interpretation of the results from the Viking mission. So you crushed that question. You didn't even know it was coming. But I see we've got a handful of questions from our listeners. So I want to try to sprinkle those in. I'll get those out and
bring it on.
Ignore mine. So a question from Mark, who asks, would there be a chance of finding a Jurassic Park DNA of an earlier Martian life form deep inside a rock structure on the red planet?
Yes. Now, of course, from the synthetic biology perspective, it would not necessarily have the same structure as exactly what we find on Earth. You might expect one of your problems with the DNA molecules that we have from ancient dinosaurs, or for that matter, ancient humans, is that the DNA is not a terribly stable molecule. It will survive for tens of thousands of years. So we can clone DNA. I mean, I've never done it, but Svante Pääbo has done it. And he won a Nobel Prize for doing it. We can clone DNA out of fossil Neanderthal bones or Denisovan bones. There have been older attempts to clone things from 22 million year old leaves from Clarkia, Idaho, for example. That turned out to not be a successful cloning experiment, because when you're trying to seek scarce DNA, it's very easy to have contaminant DNA from you in the lab and the tree outside the building give you a false positive. But the short answer to your question is yes. I mean, if the Martian DNA is not exactly like your DNA and my DNA, but is a more stable structure, it might very well survive for longer. But keep in mind that the question will be how well it is shielded, how well it is trapped. We certainly have in salt mines on Earth DNA molecules that have survived, and sometimes they're in cellular structures, which may have helped them survive for millions and millions of years.
So there's a chance. I see we have another question. This is from Bill, who asks, do we have new insights about the blue smudges in Jezero, Gale, and other craters on Mars?
So I think I even know Bill. The answer is I've tried. Bill gave me some of these pictures, and I have pondered those for a great many months. There are these little things which sort of look windblown. It looks like there's a structure which is sort of blue, which actually looks like it trails off from a source of origin in the Martian surface. I have been trying to compose an intelligent email to Bill to explain what those are, and I cannot do it. But so the answer to that question is no, sadly. But there is certainly a phenomenon on Mars that we do not understand. It's very easy to be what we call God in the gaps. Only in this case, it's biology in the gaps, right? It's very easy to say, it's something we don't understand, therefore it must be biology. But, at some point you have to establish a dialectic where the idea that it is not biology. At some point, you have to believe that Mondays, Thursdays, and Saturdays, Tuesdays, Wednesdays, and Fridays, you can believe that it is biology. Take Sunday off. But the bottom line is that the answer is no. It's but there are a lot of things like that on Mars that we just don't know how to put into our framework.
Phil was really trying to get that question answered. Yeah, had to come on here to get it.
You gotta go. You gotta go, and you gotta go with the intention of looking for that kind of answer. Now, you got to keep in mind that there are places on Mars that are more likely to hold life than others.
Mhm.
These are sometimes called special regions, and it has been NASA policy with respect to planetary protection to not go to those special regions. And so, this for those of us who are eager to know about the potential for a biosphere on the surface of Mars, and for those who are, well, anticipating in the next 2 or 3 launch cycles, Elon Musk or the Chinese National Space Station will put people on Mars. And I mean, Jan Spacek and Chris
Mhm.
Tenby, who work on the Alpha Mars. I think you've interviewed them
Yep.
in the
Yep.
past. They are very much eager to go look for life on Mars before human habitation sets
Mhm.
there, so that we at least understand we have a baseline for which we can judge human contamination. I don't think that human contamination will be all that severe all that soon, but
Mhm.
it will exist. And so I think when Jan Spacek and Chris Temby are trying to get, I mean, they have put together an organization called Alpha Mars, which is actually, you can join. It's a volunteer organization. You can join. You can put your expertise into the sort of social network that they have put together to try to encourage Congress and encourage NASA, and to sort of help the private missions to go looking for Mars, and also to raise some funds to try to define ideal life detection instruments so that we know how to look once we are able to look. Because remember, Elon Musk is democratizing launch. He's lowering the cost of launch. And it's now within the capability of doing. I mean, Jan has a thing on primordialscoop.org, which is a blog that we contribute to, which
Mhm.
is talking about having multiple. He calls it the Impress architecture, having multiple missions in the same. Going to Mars and doing lots of experiments. Each experiment individually is very cheap, maybe a couple $1,000s, $10,000, something that university students can raise enough money to. Then you bundle them all together to be able to afford the launch as a unit.
Mhm.
But then, so yeah, so all of these little things should be considered, because there's no question that the time is ripe to go now before humans go.
100%. Absolutely in agreement there. We see a question here from Daniel. Daniel asks, how hard would it be to genetically modify a plant to survive the Martian atmosphere?
Well, yeah, right. So your poor plant is not going to like being moved into an environment which has 1% the atmospheric density and a lot less water. So you got to keep in mind that Mars has elevation. Jan has taught me all about this, right? There's the Hellas Basin, which is a big depression. Roughly, if you were to draw it on the map of the United States, it would fit in sort of the Midwest, going from the Texas up to the Canadian border. But it's low elevation, higher pressure, therefore warmer, much more liquid water. So I would try to get a plant now from Earth, from someplace that's high up and cold,
Mhm.
because this would be the closest analog. And then we would try to modify it. But, that would be a very hard job to do, because the plants have themselves evolved to handle even the attack in the desert, where the pressure still is much, much higher than it is even at the highest pressure on Mars. So you would have to work very, very hard. So I would start, frankly, not with a plant, but with a photosynthetic microbe. There are algae that are growing. There are also bacterial that are photosynthetic. I would start with a bacteria which is photosynthetic, a cyanobacter. I'd give it to Lynn Rothschild out at NASA Ames. She is sort of the synthetic biology guru there. And we would try to put this poor organism under selective pressure, where it's forced to live with less water and less pressure and colder temperatures, and see what we could do.
Mhm. Okay. Okay, well, speaking of that, question from one of our ambassadors, Stuart. His question is: it's been almost 50 years since we sent a biology experiment to Mars. What would you choose to send, and what would you test for?
Okay. Well, you're walking squarely into my trap. Yeah. No, no. We have worried very much from synthetic biology perspective as how we would look for not the products of Darwinian evolution, but the molecules that enable Darwinian evolution. One of your problems in biosignatures, if people go to Mars, there are these experiments that have looked for amino acids. Why? Well, because we have amino acids. But the problem is that meteorites have amino acids. There are many ways of making amino acids that do not involve life. And so, and that's true for almost every other product of Darwinian evolution. So Lee Cronin and Sarah Walker, of course, try to go into very complex molecules. We can try to look for things that are so complex that we have to believe that they must be the products of Darwinian evolution. But every time you can suggest one of these complex molecules, it turns out that, well, hey, they can also be made by non-biological processes. But one thing that really can't be made by non-biological processes, at least not kept around for a long period of time, are the genetic informations, these DNA-like molecules that are not products of Darwinian evolution, but they're necessary to implement it. And one thing that synthetic biology has taught us, that when we have molecules that are DNA substitutes that work versus ones that do not work, one of the characteristics that make the molecules work, that distinguishes from the molecules that do not work, is that they have a repeating charge on them. In DNA, it's a repeating negative charge. You can imagine a repeating positive charge as well. But these are called polyelectrolytes. Of course, an electrolyte, which is what you drink, is a salt with a positive and a negative charge. But polyelectrolytes are where you have the same charge all along a backbone. So you have, DNA molecules. If it's 1,000 nucleotides long, it's got 1,000 negative charges on it. So as Jan has pointed out, it's very easy to recover polyelectrolytes from water. All you have to do is put them in an electric field. You got a plus charge over here, all the negative polyelectrolytes will go to there. And if you have all minus charge over on my right hand, your left, all the negative charge, positively charged molecules will migrate over here. So if you can set up an electrodialysis experiment, you will be able to recover polyelectrolytes from very dilute solution. So one of the assumptions is that in this near surface Martian ice, I mean, Jan loves to call it lasagna because it's a layer of ice between a layer of Martian dust and a layer of ice and a layer of Martian dust, right? You ought to be able to melt that ice. Elon Musk, SpaceX, is planning to melt that ice to generate raw materials, a resource to make propellant for the astronauts to come back to Earth. So if you put an electrodialysis on a stream of that water, the idea is that you'll be able to recover sparse, scarce polyelectrolytes from sparse, scarce life in this near surface dust, ice, lasagna, and then we'll be able to detect that. Now, those molecules will have to have another property. They not only have to have a repeating charge in them, but they also have to have a relatively small set of size, shape, similar building blocks. So after you concentrate them from the Martian lasagna, soil, ice layers, you have to now look at them to make sure that they're being built from size, shape, regular building blocks. And that is a biosignature, not of molecules that are made by Darwinian evolution, that have the complexity of Darwinian evolution, but rather they're necessarily to enable Darwinian evolution. You have to have those. And you'll have a molecule with has those two properties, a repeating charge and size, shape, regular building blocks on Vulcan, Kronos, Tatooine. My gesture, I just managed to knock my microphone, so if you lost it, let me know. Yeah, and so this is the kind of thing that I would look for tomorrow if I had a ride.
You would look forward tomorrow if you had a ride, is what you said?
Yeah. Yeah, so I would send a machine to Mars to do electrodialysis to recover biopolymers essential for Darwinian evolution from water, lasagna, ice, dust, lasagna on the near surface of Mars, especially if it were to be generated and delivered to me in large amounts by Elon Musk mining that water to make propellant in preparation for the arrival of humans.
Okay. Okay. This episode for sure takes the cake for the amount of times I've heard lasagna.
You
I do
take the
remember.
lasagna. It could be a layered cake, but Jan, for some reason, likes Italian food, not Bavarian
I do,
chocolate cake.
I do remember him mentioning that. Yeah, when we got to connect. Let's see. I know we've got a couple other audience questions. Let's see. Is there any sample from Mars sample return you are particularly excited to examine? Maybe comment geyser.
Well, I'm a little disappointed about our sample return. We're having a hell of a time getting these samples back, and um,
Mhm.
a second problem is that they're also being collected from a not special region. So they've gone to a place where they're looking for past habitability. That was part of the mission design spec, and perhaps a past evidence for a past biosphere. My buddy Martin van Kranendonk, who is one of the guys who is a curator of these very ancient fossils in Australia, has some silica structures which look very much like hydrothermal silica precipitated by biological molecules. I would love to look for those inside of the sample return. But the problem fundamentally is that the planetary protection community has been so concerned about the sample return that the planetary protection steps have driven up the cost of the sample return as to make that sample
Mhm.
return not very likely.
Mhm.
My story on that is, of course, that Robert Zubrin, whom you know very well, has estimated that maybe 500 kilograms of Mars come to the Earth every year by impacts on Mars, injecting stuff, which then wanders the solar system before landing on Earth. And now I've got 5 grams of Mars sitting out in the outer room in the museum, which was Bob Bruner, who's a layperson in Denver, actually helped us buy. A few kilograms of Mars brought back from NASA is not going to change the calculation as to how much Martian exposure we have to a Martian biosphere. But the problem would be that if I had were going to do sample return, I would have gone to a place that's more interesting. I would
Mhm.
understand that the problem of reverse contamination in terms of hazard is negligible, and I would have the samples back much faster, much lower cost, and then have a much more interesting sample. But you got to keep in mind that the 2013 decadal survey, which is what
Mhm.
NASA does every 10 years to try to
Yep.
define space missions, made this decision that they did not know how to look for life on Mars today. They had not read our papers. They did not understand completely how badly the Viking results had been misinterpreted.
Mhm.
They still thought that the surface was self sterilizing. And so they decided that there was no extant life on Mars. We could at most hope for ancient life. You have John Rummel, who is, had the world's greatest title. He was director of planetary protection for the United States government. How would you like to go home at night and tell your kids that you were the director of planetary protection? So Rummel and Conley had a paper in 2017 where they said, look, the community has not been persuaded that the search for extant life on Mars has a high priority. And in our view, that was a misinterpretation of not only the work from synthetic biology, but also the understanding of Viking that was emerging at the time, actually had emerged years earlier. And so we got saddled into a very expensive sample return mission, made even more expensive by what we think is a misplaced
Mhm.
concern about bringing life back to destroy humans or the ecosystem or plant life or something.
Right, and we've seen Mars sample return obviously be delayed,
Yeah. Yeah.
over budget, beyond timeline. So right now we don't have a status on that. I think the most recent was a call for abstracts on potential solutions. So maybe we'll see those samples come back.
But
Uh,
those solutions are going to cost money. And if you insist on bringing these things into an orbiting platform where they can be quarantined in orbit for, whatever, the engineering analysis will say, okay, well, what are the chances that that mission fails and the rocket doesn't go to the orbiting satellite, but rather lands in Antarctica or lands in Gainesville for that matter? And the answer is, well, it's a 1 in a million chance or 1 in 10,000 chance. There's always some chance of mission failure. And
right.
so that basically, as you try to drive that mission failure chance down to zero, you end up it's exponential in terms of the costs that you spend. And this basically planetary protection means that for sample return, that basically no science is going to get done at all.
Mm-hmm.
I mean,
Yeah.
I was in 2000, even 24 years ago, I was in a planetary protection meeting that John Rummel had organized. And I mean, the only thing memorable about that meeting was I got the worst case of influenza that I've ever gotten. But we spent a lot of time trying to figure out all the possible risks, one of which, is you would have ice 9. I don't know if you're not familiar with Kurt Vonnegut, but we would have a form of calcite that would come from Mars to Earth and reseed the crystals of limestone that are all calcite and aragonite that are all around the planet, and have the entire Florida limestone caves collapse into rubble. Okay, so now that means you got to dissolve the Martian rock in hydrofluoric acid. So at some point, you're so much treating the sample to prevent a hazard with negligible likelihood from occurring that you end up making the sample not worth having, even if you could return it.
Right. Yeah, that leads to a good question from Greg here, who asks, what's the basic reason NASA has refused to put life detection on probes for 50 years?
Okay, I have an opinion, and this will be an opinion that will be hotly disputed by Mike Meyer at NASA headquarters and other people. Maybe, I don't know. I've not asked him lately. But look, in 1976, Brian Dobbs, who does a documentary, he has a documentary called Blue Planet Red, which is a discussion, among other things, about how when Percival Lowell, there you are back with that name that we started the conversation, had persuaded the culture that Mars was alive. Then Mariner went by in the 1960s and found that there was no atmosphere and no water, looked like the moon. So Mars was dead. And then Mariner 7, or whatever number it was up to, by then went back and discovered that actually there could be life on Mars. You could see rivulets of water and all the rest of it. Mars was alive. And that was the culture which put Viking on the surface. And it was a very expensive mission, right? It was running duplicate. The GC mass spec at the time was perhaps the most expensive scientific instrument it had ever flown. But then what happened when 1976 came along and the GC mass spec said there are no organics, and Jerry Salfin said that's the ball game, no organics, no life. The whole program shut down. Doors were locked at JPL in weeks. People were fired. They went on to other careers. Nothing in government shuts down. Okay. So when you have a government program shut down, that's quite traumatic.
Mm-hmm.
Right. So I think that the NASA culture, now remember, NASA is a logo, a building, a couple people, right? NASA is basically takes its cue from the community, right? So NASA is really very much an outward looking organization. So NASA itself doesn't make actually decisions. It makes decisions, but it makes decisions with a huge amount of community input.
Mm-hmm.
But when the culture of the community says that Mars is dead, no one's going to recommend going to Mars. From 1976, 1986, 1996, 20 years went by with essentially no missions flew. Then, of course, Dave McKay came up with this Allen Hills meteorite, which had little structures in it, which people interpreted as fossils from Mars. This brings Bill Clinton, the president of the United States, to the Rose Garden for nothing brings Bill Clinton to talk about science. You can cure cancer, and the best you'll get is Tony Fauci or the National Institutes of Health head Francis Collins will come to the Rose Garden. But the president comes to the Rose Garden when you discover alien life. This is what people are interested in.
Mm-hmm.
Now, the point was the story that I've heard, I don't know if it's true or not, was that Clinton went to Dan Goldin, who was the head of NASA at the time. I've tried to get a hold of Dan. He's still around. I tried to ask him whether this story is true, but Clinton allegedly had books in the Oval Office on Mars spread out, and he asked Dan, "Is this life or isn't it?" And Goldin says, "I don't know." And Clinton says, "Well, why the hell are we paying you? I mean, NASA is the organization that the U.S. government has, so that when we encounter little green men, right, we can have an expert." But Goldin was an excellent administrator, and he managed to get the Astrobiology Institute program starting in 1999 for 20 years, funded. A lot of good science has come out of that. But we were told in 1999 that we're not doing explicit life detection missions. We will follow the water. We will look for habitability, but we will not look for life because, and then this was the subtext, we believe, the culture believes, that if the community, the public, which pays for all this stuff, right? The most important thing in science is money,
Money.
right? If the society is dissatisfied with ambiguous results, and we make our mission finding life, we're destined to get ambiguous results, and therefore we're going to undermine the enthusiasm of the community for the enterprise searching for alien life. Now, I think that does not give the community enough credit. But the idea was we could say we're looking for evidence of water. We can find that. That community, the public will be satisfied. We can go look for habitability. If we can define that, we can find that. The community, the public will be, the taxpayer will be happy. I think that underestimates the sophistication of the American public. I think that they are perfectly able to understand that life is an ongoing process, that science is perhaps defined as a process by which error is managed. That's what science is. It's not hypothesis testing, as you learn in your 8th grade science fair project.
Right.
It's a way of how you intellectually, in a community, without a judge, without a gatekeeper, right? There is nobody who sits atop the structure saying this is true and this is false. It's a community dynamic, crowdsourced, dispersed activity, but it's a way in which you manage error. And so I think the community can sort of understand that. And I think we saw that during COVID, for example. There was a lot of cases where the science made some pronouncement, but the public is perfectly aware that you can't say you should be standing 6 feet apart in a grocery store. It could be 7 feet, it could be 5 feet. The public is perfectly aware of saying, well, how the hell could they possibly do an experiment that says it is 6 feet, not 7 feet, not 5 feet? The public is educated. The American taxpayer is relatively sophisticated. But I think the answer to the question from the chat is that the reason why NASA has not made explicitly their goal of searching for extant life on Mars is because they are afraid that the public would not accept an ambiguous answer.
I'm going to have to agree with you on that one. Yes, I think to end things on a positive note, like you said, the American taxpayer is educated and they don't want an ambiguous result. So more reason for us to...
No, be careful here. The taxpayer is educated and they understand and are willing to tolerate the fact that the science does not give them an unambiguous result initially. They are prepared to go back again, and they would be going to go back again. And the only point here would be that and I'm sure they would have gone back had the Barsoom model for Martian life been introduced in 1976 as opposed to 50 years later. There would have been another mission constructed, and Gil Levin Chris Temby tells me he loves the chiral label release experiment. That would be his next job. But the point is that there would have been a follow-on experiment. They would have gone back with a better set of life detection, and they would have perhaps given an ambiguous result as well. And then, so the community, the taxpayer, is not demanding, in my view, an unambiguous result. They are looking for progress, and they understand that science will not necessarily give them an unambiguous result initially. They would like to have one eventually.
Right. More reason to continue testing, researching, to get those samples back so we can determine some of those answers. Well, Dr. Benner, I want to say thank you so much for joining the podcast today and answering all the questions. You rallied. There was quite a few in there. So thank you for tackling all of those. And it's been a pleasure to chat with you.
It's entirely my pleasure.
I want to say a quick thank you to our executive director, James Burke, to our PR director, Michael Stoltz, our friends at Liftport, Michael and Leah. And thank you, everybody who tuned in for today's conversation. Like always, we're going to wrap up today's episode with the same phrase you've heard me say before.